Good evening, ladies and gentlemen. I would like to begin in our customary way by acknowledging the Wurundjeri people of the Kulin Nations, traditional owners and custodians of the land on which this event takes place. Welcome, ladies and gentlemen, to this very special public lecture, Minds on the Markets, at the University of Melbourne Faculty of Business and Economics. We are truly fortunate tonight to hear from Professor Read Montague. Read is visiting us for the Melbourne Brain Symposium. You may be wondering how the Brain Symposium and the Faculty of Business and Economics could possibly have a connection. So let me try to explain in as brief a moment as I can. A veritable revolution in the study of human decision-making is taking place. This revolution will allow us to better understand emotions, intuition, intellectual discovery, risk-taking, crowd behavior, and actions in relation to exuberance, panics, and crashes. We have come to this point due to advances in neuroscience, where we can now take detailed measures of brain function and connect that to crucial aspects of cognitive function, as well as advances in computer science, which now provides not only the right insights on how to think about difficult problems, but also provides us with the tools that can help us do better. Our goal is to bring this decision-making revolution to Melbourne. As Dean of the Faculty, I am delighted to tell you that we are embarking on an extraordinary series of initiatives that lie at the intersection between human decision-making, neuroscience, and machine decision-making. We are building an interdisciplinary team of decision scientists with backgrounds in economics, finance, psychology, neuroscience, computer science, and related fields. With Professor Peter Bossaerts, a global pioneer in the development of neuroeconomics and decision neuroscience, recently joining the faculty from Caltech, the University of Melbourne now sits at the cutting edge of this innovative research area. An example of this innovation is the Brain, Mind and Markets Laboratory, which Peter established along with my colleague, Dr. Karsten Murawski. The lab conducts experimental research on individual financial decision-making and on financial markets. It studies risk decisions all the way from the brain to markets, integrating knowledge from decision and game theory with neurobiology, economics and finance, psychology and computer science. Having this expertise right here in Melbourne makes sense. Informed decision-making lies at the heart of a number of economic issues, including superannuation and retirement savings, the costs of obesity and pathological gambling, and equally, Melbourne's pivotal role as Australia's financial services sector means that industry can truly benefit from those new insights and accomplishments that arise from this coming revolution. And so it is only appropriate that we welcome tonight Professor Read Montague to the faculty to deliver this special lecture, Minds on the Markets. I would now like to invite my colleague, Professor Bossaerts, to introduce Professor Montague.
Thank you, Paul. It's a great honor and a pleasure to introduce Professor Read Montague to you. He is the Vernon Mountcastle Research Professor at Virginia Tech, Director of their Human Neuroimaging Lab and their Computational Psychiatry Unit, Professor of Psychiatry and Behavioral Medicine, and Professor of Physics. These titles are telling. Professor Montague occupies an enormous space in the building of science, engineering, and medicine, from mathematics over biology, psychology and economics to psychiatry and physics. Indeed, Professor Montague is the 21st century equivalent of a Leonardo da Vinci, a person eager to understand and to create, no matter where the application is. In my personal experience with Read, you throw a problem at him and he'll solve it. In fact, that doesn't give him the right credit because he sees the problem before anybody else does. as he recently proved when he started developing new electrodes to record changes in neurotransmitter concentration in the human brain. That is to do what neurologists call voltammetry. In fact, we will talk about this tomorrow at the Melbourne Brain Institute. Professor Read Montague is the pioneer of decision neuroscience. And decision neuroscience is a new branch of cognitive neuroscience where one uses the formal language of control theory, of machine learning, and of economics and game theory in order to decipher the computations in our brain that explain why we choose what we do. That is, the neural algorithms behind human choice. One cannot overestimate the importance of this. Our choices are driven not only by cool calculations to attain certain goals, but also by elusive things such as emotions, intuition, and impulsivity. For years, neuroscientists therefore thought that the parts of the brain involved in determining choices, the prefrontal cortex, the subcortical structures, that is your rat brain, and the brainstem, your reptile brain, could not be understood formally. That was in contrast with, say, for instance, the visual cortex, where mathematics had been used most effectively to understand how we see. For the first time, it became possible to formally explore elusive aspects of human condition, emotion, intuition, trust, creativity, and in financial markets, in the financial applications, greed, exuberance, and even bubbles and crashes, as you will probably hear about later tonight. Rather than going in detail over Professor Montague's many accomplishments, including positions, prizes, and publications, I actually prefer to end my introduction here because anything Professor Montague has to tell us is far more fascinating. Read, we are ready to listen. You have the floor.
Wow. Those are quite some introductions. I'm very pleased to be here. I'm slightly jet lagged. I'm happy with the turnout. I'd like to see one show of hands. People here who are not professors or students that want to be professors, just to give a sense of it. Okay, so this is I was told this would be a mixed lay audience. I assume that's a compliment. And so I'm going to take you through some things. First, I'm going to tell you what it means for me to be a computational neuroscientist and the point of view that that brings to looking at the brain. And then I'm going to segue into a couple of examples I think you'll find interesting, if not horrifying and entertaining. Okay, so point one, it seems redundant. What's the relationship between the brain and the mind? This is something that people like me are very interested in. People that study the mind are interested in mental functions, capturing them in mathematics and descriptions. And people that study the brain are interested in the cells and interactions about molecules that mediate the electrical activity in your brain. The question is, how does the one provide support for the other? A second question is, why does it even matter? Okay? And there are people that think that. There are people in the academic world who think that's a distinction that doesn't matter at all. Why can't you just study as what's called functionalism? Why can't you just study the mind and never worry about the implementation in the brain? People like me who have, as you saw, lots of jobs and titles, that's job security. I'm over 50 now, and so once I lose one of those jobs, I have another one to replace it with. I think it matters because you're talking about an organ that's been evolving for billions of years, really, if you go way back. There must be some tricks embedded in it. It's highly energy efficient. Your brain runs on about 23 watts. Your whole body runs on about 85 watts. If you're in the Tour de France and you're climbing one of the mountains, a trained cyclist might peak at 400 watts or something like that. But as you guys are sitting there, your brains are running on about 23 watts, and that's being distributed to everything that your brain does, all the functions it does, keeping your body alive, thinking, and all this stuff. We still can't mimic some of the things the mind and brain can do, and so that's a freakishly efficient device. So people like me, and now people like Peter, are coming together to look at events of the mind that are most interesting to us. Markets are interesting to everyone, and events of the brain and how the one explains the other. Okay. One of the key concepts I'm going to talk about is what's called the computational theory of mind, and I'll bring some detail to that in a minute. We don't mean the brain as literally a computer like the one on your desk, but we do mean you can describe the brain and the things it does in terms of computations that it emits. And you can use those descriptions to try to connect those computations to the ones that you would have to be doing to account for mental events. Okay? So that's coming. So this is not a relative of mine, but it could be. So this is the universal sign in America for choking. I am choking. If you go into a restaurant, there'll be signs on the wall, and it'll say this. You teach your kid to do that. But you guys recognize that or something like that immediately. I don't have to tell you anything. The inference is automatic. You see that guy doing that, you see him doing that in a restaurant, you might even run over to help save him. I don't have to teach you that. So why is this a universal sign for thinking? Ever thought that? This is a sculpture by Rodin. We've all seen various versions of it. The other thing is, as I was preparing this talk last night, I was sitting in my chair and I tried to do this position. And it's an incredibly odd position. His left hand is on his left knee, and his right elbow is bent across his body, also on his left knee. If you do it now, you'll go ahead and give it a go. Give it a try there. Yeah. It's hard to do. You have to contract your. I think he's working on his external obliques. So I don't know the answer to that. I don't know why this is a universal symbol for thinking. But we all know that it's a universal symbol for thinking. We make deep inferences like this on scant data all the time. Okay? How is it that your brain gives rise to that? This guy, does anybody recognize this person? Anybody shout out his name? Alan Turing. This is the thinker of the 20th century. If I were going to give credit to the three biggest scientific advances in the 20th century, one would certainly be computing. This is the father of computing, of the idea of modern computing, Alan Turing. He's now famous because they made the movie about him and his contributions to World War II. But he worked in lots of areas. This is actually right before he was, he killed himself. He also worked in morphogenesis and embryogenesis and other areas of biology. And he was an amazing guy who I don't think ever really got credit for that. The modern notion of computing is due to Alan Turing. And I've drawn a depiction here of a Turing machine, which was an imaginary device that he made that had a tape. You could write ones and zeros on the tape. It had a reading head that could move one direction or the other and read or write from that tape. And he showed that anything, well, actually all the machines that you use, unless somebody has some special device in their pocket, are Turing machines. That's the modern idea of computing. It evolved out of work that had been going on in the decade prior to that, work on something called the lambda calculus, but he's the first one that translated it into something very concrete and easy to get your mind around. He also came up with what we now call the computational theory of mind. And Turing's idea was this. Back in his day, a computer was a person. You had rooms full of computers. They were people that computed things for you. A computer was a person who sat there and did computations for you, made actuarial tables, computed things for railroad timings and whatnot. Now we don't think of them that way. And what Turing said was he took on the fact that the brain and the mind is like a computation. And since it's like a computation, maybe we should make one distinction. Now to us, this is an absolute commonplace idea, that the hardware and the physical interactions in the hardware that support the computer is different than the patterns of information processing running on it. The program is different than the computer that supports the program. In fact, programs now, when you load them into a computer, can be relocated anywhere in the computer. They're busted into parts. The parts talk to one another. And so where the program is not something that's necessarily constant when it's running. When you have a program that's controlling the cooling towers of a nuclear power plant, it's performing operations and controlling variables, and these variables have dynamics, and those are separate from the physical instantiation of implementing the instructions in the computer. So what Turing said was, we have to distinguish your brain, the hardware, from the patterns of information processing that's your mind. Okay? That's the computational theory of mind. And he did just what I've written right here. In a sense, he proposed a very concrete answer to an old philosophical problem. How do you get mind-like stuff from stuff-like stuff? Yeah, well, I tripped up Descartes. So the mind is not equivalent to your brain. It's the information processing supported by your brain. And the metaphor that you should carry away is it's like software. So there's a sense in which you could have something wrong with your software and have no identifiable thing wrong with your brain. Okay? You could just be updating variables at the wrong rate or in the wrong place. You could be moving them to, a location where they're not found in the right way. You could be indexing memory in a way that wasn't quite right, or the timing was a little bit off. But none of the parts of your brain are broken. Okay? That's a case where pieces of your mind would be broken and your brain would be completely intact. It can also go the other way around. Whatever you want to call your mind is running on a brain that's always replacing its parts. You replace every piece of your soft tissue in your body every 2 years. It takes about 11 to replace your skeleton if you have a fast turning over skeleton, maybe 13. Okay? So every 2 years, you're looking at a completely different set of atoms constituting you, and you don't forget who you are, and you don't forget what you like, whether you like ice cream or chocolate or something like that. And so you're sort of an eddy current of matter flowing through you, organized by your cells and your DNA and the body that's been built. Your mind's the same sort of way. Your mind's flitting out, it's running on parts that are dying and replacing themselves and whatnot, but you don't feel that. There's a continuity to your experience. That we don't know how to explain. Computational theory of mind. So that's the prevailing ambition for people like me, is that maybe we can describe events in the brain, the hardware, as sets of computations, and events in the mental and behavioral space also as computations, and then push these two parts together to see how the one explains the other. Okay? It's easy to say, and it's hard to do. The reason it's hard to do is there are many, many levels here, all the way up from the cells that compose your brain and the biochemical networks that live inside cells, up to the way individuals compose themselves into groups and collections of people and institutions, and then culture gets built out of that, et cetera. Okay, so it's an easy problem to talk about, it's a hard problem to do. So you have to kind of pick a level. You also have to note that these levels are real. You can't pretend like they're not, like they're there, like they're not there. Let me just give you one little, this is my neuron slide. I am a neuroscientist, so I have to have one neuron slide. This is my one neuron slide, just to show you and give you a sense of the scale of the structure. So let me draw your attention here. This is a pyramidal cell in your cerebral cortex. The width of that cell body there, that's where the nucleus is with the DNA in it, is about 30 millionths of a meter across. Okay? These are called dendrites. These are processes that grow out of the cell. They receive inputs from other cells. Those inputs are magnified here, come to affect the cell through things called synaptic junctions. Synaptic junctions are plastic, meaning depending on the experience that you have and the electrical chemical activity there, they can change their strength of the connection. They're complicated. Okay? They're about a micron, they're about a meter, 1 millionth of a meter across. And in a cubic millimeter of tissue, about the size of a match head, there are about 1.5 billion synaptic connections, at least in the human brain. Okay, 1.5 billion, it's a little tiny space like that. And there are about 3 miles of dendrites, and there's about 1 mile of axon. Now, an axon is also a process like the dendrite here, and it connects one nerve cell to the next nerve cell. All right? All of these parts are turning themselves over and replacing themselves all the time. They're putting proteins in the membrane, parts become senescent, they're replacing biochemical networks, et cetera. So there's an enormous amount of miniaturization that goes on in your brain and dynamic replacement of parts that we really don't appreciate yet the subtleties of what it's doing from a computational point of view. Okay? So there's an enormous amount of structure down here at the cellular level that we can study. We can study this. Neurobiologists here study things like this. We don't yet appreciate the subtle connection between that and features of our mind. But what we are talking about here, and what Peter Bossarts has been brought here to, I think, catalyze, and what the dean was enthusiastically endorsing at the first part of the talk, in the introduction, was that now in the decision sciences, and in particular in the decision sciences, we're seeing a way to connect up ways of modeling behavior that are new, that are different than the things that experimental psychologists did 50 years ago, and giving new insights into how to use that to model parts of the brain. Okay? So why should economic models provide any kind of platform for understanding the brain? I'm going to give you my 3 favorite reasons. Why did economic models attract me? Well, I'm a sort of mathematical neuroscientist, so if you can write it in an equation and use fewer transitive verbs, then I'm a happier camper. But that's just my sort of sensibilities. But the main reason is they're normative. So economic models, whether they're right or wrong, they're normative. Instead of just summarizing data by looking at my nervous system and sending information into it and asking how my nervous system reacts, that's the way an engineer would analyze it. What an economist does is they say, what should the organism do? What should it do? And there's a concept of rational actor there. What should a rational actor do if they were trying to decide between option A and option B? Okay, that's a different kind of perspective. So instead of providing models that just summarize the data that you see in an experiment, it provides a model that guides the structure of the experiment. You go looking for the way in which the brain is doing something normative. The second thing is they're sort of mathematically committed. They're crisp and clean. And the variables that do work for describing behavior in the brain, you can see them. They're not just woolly concepts. It's a crisp way of describing things. And the thing that's most interesting is they extend beyond individuals, right? Economic models extend beyond an individual agent to institutions, markets, schools, the way components of culture interact, because those components affect the way you perceive the world, all right? There's no provision for anything like that yet in neurobiology, but this is growing up. All right, I'm going to give you 2 examples tonight. And what they're going to do is they're going to lean on another revolution that's taken place, which is non-invasive brain imaging. I'm going to make some reference to the kind of brain imaging I'm talking about. And at the end of the talk, I'm going to point to the future of where brain imaging is going, the new technologies that are on the horizon. And we're going to address 2 things. 1, market bubbles, and 2, political ideology. And by political ideology, I mean conservative and liberal, not party affiliations. And I don't know much about Australian politics, but in fact, the experiment I'll show you was inspired by work in a twin registry here in Australia a few years ago. Market bubbles are an important aspect of all our lives because just let's dial the clock back to 2008 and ask, although I don't know that Australia was rocked the way the US was, it affected everybody's life in the US. It affected the institution I was at. I'm at Virginia Tech now because of an economic blow-up that went on in 2008. So not understanding why markets blow up the way they do, all kinds of markets. Artificial markets we have now where we use to invest in equities and vote for one company over another, those are good markets, but markets have been around since biology's been around, okay? Okay, here's the problem. And I've listed it as a question, wisdom of the crowd. So this guy is asking for a vote yay or nay from those, and you see the problem here is that thought bubbles are private, right? We don't know when everybody says yes, what they actually mean and what's living in their heads. A market, at least in my understanding of it, is a good mechanism, certainly for doing an experiment on a group of people like that, an experiment that can do 2 things. One is that it can extract some of this private information and put it into a public domain, and 2, compile it together to produce something that reflects something more than what each of these guys knows. In that case, it'd be the price. Okay? So market is a great simple example. It's a great experimental platform for a neuroscientist like me to look at the way you can take information out of individual people's heads and combine it with a mechanism that's not human, a way of making a book and making a price, amalgamating that together and sending it back into the heads and asking how, when you rinse and repeat on that cycle, how does that work? I'm particularly interested in what happens when people overvalue the equities that they are investing in. What happens when you have a bubble, okay? Here are a few famous bubbles. My favorite one is the green bubble here. This is the 1929 crash of the Dow. Make of it what you want. It is, people argue about whether bubbles exist. There is not universal agreement on this. And I know there are economists in the audience, so I'm not going to say anything particularly technical about bubbles, but I am going to show you a bubble experiment. This is what we're interested in understanding. No more to say about that. They're hard to identify. The experts and the experts in charge of the experts, some of them quoted here, can't really agree. In fact, Bernanke, who was at Princeton when I was at Princeton, he's an expert on the 29 crash. And he has a lot to say about it, but none of it's neural. Okay, so we're very interested in what's going on in your brain when a group of people is overvaluing something, and we think we've pinned down a lot of the other contributing variables. Here's what we do. We use a laboratory market paradigm. In our particular case, we took a paradigm from another group that had published a paper in 2005. I won't go over the details of it. I'll just highlight the high points. We measure neural activity using multi-subject functional MRI. I'm going to tell you what I mean by that. Has anybody not heard of functional MRI? Can I see a show of hands? Who has not ever heard of functional MRI? Of those who have not heard of functional MRI, this is a marginal. Have you not heard of MRI? Everybody's heard of MRI, right? Okay. So an MRI, here's one here. This is a head-only MRI. Uses magnetic fields and radio waves to take a snapshot of like your knee or your liver or your brain. And these are typical images that you've either had made yourself or you see them in television shows and in advertisements. You get these gray level images, and they're static snapshots. About 1989, it was demonstrated that you could use MRI in a different mode, and instead of making snapshots, you can make microscopic blood flow movies from thousands of points in a brain at one time. Okay? So instead of just using this machine to take a snapshot of my brain, I could use the machine to take, let's say, 50,000 snapshots through time of microscopic blood flow changes. Okay? So that's pretty exciting, right? Here was my Alaska trip. Here is my brain blood flow movies. The reason that's important is the brain is efficient, and there's a tight coupling between neural activity changes in the brain and local microscopic changes in blood flow. So if I have a proxy that can measure microscopic blood flow movies, then I have a way to measure changes in neural activity. Okay? So you lie someone, it was the first time that we'd had a method that could give any spatial precision to measuring brain activity in a healthy human being. The only way that we can measure things with that kind of spatial precision in a healthy human brain is to stick a micro wire in your brain, not something most people want done. And it's done during neurosurgery when you're looking for an epileptic focus or where you're planning for something. And so only people that were already there for pathologies or clinical intervention could we do these recordings from. This is the first time that was true. It's created a real revolution in cognitive neuroscience because you can now take healthy people, put them in these machines, make them dream, have them think of their sailboat or their bunny, have them look at awful pictures, have them do motoric acts, build boards up where you have dancers that dance on the boards while you're doing it. So we can study all kinds of things that we wished we could study before. It's a slow method. It doesn't, you have to design experiments where things are happening kind of slowly because the blood flow responses are relatively slow. And so we're talking about being able to measure events in your brain that are going on in the order of seconds to tens of seconds. Okay, so that's not everything. We wish that we had a lot faster methods with this kind of spatial precision. That's coming, but that's not here yet. Okay? Functional MRI. Multi-subject functional MRI is something that we do. We have created software that'll take and link up these kinds of brain scanning devices, which exist at every major university and clinical center in the world, and synchronize them. So we can synchronize signals between these machines wherever they are over the internet too. That allows us to study interacting brains and simultaneously eavesdrop on their brain activity. We call that hyperscanning. We've been doing that for about a decade. I think we're still the only group that does that. It's a little hard. Here's the experiment. We have 20 people online at once. This is an experiment that took place in 3 cities and lots of different rooms. So we were using a behavioral lab at UCLA where we had 16 people. We had 2 people in scanners in Roanoke, Virginia, all the way across the country. And then 30 miles away, another person in our 3rd scanner on the main campus of Virginia Tech. And then we had extra people in behavioral booths that were near our scanners at Virginia Tech. So we have a whole amalgamation of folks. The software takes that and keeps everything synchronized. And they're playing a simple game. This is the game they're playing. They're playing a market. It's just what you think. You start with some money, and during any round in the market, you've shown how much you have. You have an equity. Some of your money is in cash, and some of your money is in stock. Each round, you can buy, sell, or hold. You can buy or sell 1 unit. Okay? So you can't just go rushing in and throw all of it in. You can buy or sell 1 unit each round, or you can decide to hold out. Each round in the task, we do something that's called eliciting a micro demand curve from you. So what we do is you sit there and we say, would you buy for this? Would you sell for that? Would you buy for this? Would you sell for that? And you answer this question 4 or 5 times. And we zoom in on where you are. We create a demand curve, and so we have a supply and demand, and we set the price in the way markets set the price. It's, there's some technical terms to describe that. I'm not going to go into that because it's not the main point here. But you get the point. Price is at something. Now you decide, are you going in the market, or are you going out? One of the novel features of the design here, and this shows you what the outcome screen is. So you put your orders in. The orders are matched up. The buy and sell orders are matched in the market, as they would be. And the outcome is shown to you there. What I'm showing you here are traces from 16 different cohorts, averaging 20 people apiece. On the vertical axis here is the price, and the horizontal axis is trading round. Okay? So with 20 people, you have to wait for everybody to synchronize. That is, everybody has to finish submitting all their orders. It synchronizes, the price clears, you see the new price, then you're asked again, buy, sell, hold. Would you buy for this? Would you sell for that? Okay? The unique feature is that the fundamental value of the equity, of the stock that you're trading, is flat. And you know that. It's made transparent to you. I'm not going to show you how it's made transparent, but you know it. It's 14. And I've drawn that right here. That's 14, all the way across. That never changes value. I'm squeezing too hard here. So here's 14. One of the things you notice is if you follow any of these price traces, like follow this turquoise guy here, it comes up, it goes down. This one was up, it comes down. They're all getting out before the end of the market. So they feel the end coming. They know there's 50 rounds in the market. They know what round they're on. All right? So they feel that. They feel that coming. So that's, we train them on this so before they play the game, so they know it's coming. Lots of bubbles form. So remember, the value of this thing when you finish is going to be $14, and that's way over $150. Okay? So that's a big bubble. That's really overvalued. What we're interested in is, what is your brain doing when this thing is running up? One of the areas that you would look at in the brain, and there's lots of things I could tell you, but one area you would look at is a region called the Nucleus Accumbens. I'll apologize now for the names of regions in the brain. It's not my fault. It was 19th century anatomists' fault, and we just inherited all that. They're goofy. They're all derived from Latin terms that mean, the island sitting here, the area in between the sky and that. I mean, it's just, it's not very systematic. There are lots of naming systems for the brain. I apologize. So we're going to look at a region here and here. This is called the ventral striatum. So we're going to slice through your brain like this. This outer edge here that's in gray, going around like little leaflets here, that's called your cerebral cortex. This region here is called the striatum. These structures here are called the striatum. They used to be thought to be involved mainly in the selection and sequencing of actions. Now we know they're deeply involved in the valuation and choosing of alternatives, including planned alternatives, imagined alternatives, etc. They're also involved in the processing of rewards. So this region right here receives inputs from your brainstem that provide a lot of information encoded about rewards and what will be rewarding and what might be rewarding and models of what you had expected. Okay? We're going to look specifically at that part. I'm just going to go ahead and put my little arrow on here. So here's my picture of the brain again. Remember, this is called a coronal section coming through. Here's the nucleus accumbens. And what I've done here is I've shown you the rounds before and after the peak. So I've taken all those markets and I've scaled them down and lined them up so that the peak of the markets are here in green. And what I'm showing you in blue is the activity in the nucleus accumbens. And because of the way I've plotted this, that blue curve is shifted slightly to the right. This is a 5-round running average. And so if you look at the peak here, it would actually be just before the peak of the markets here. So what you see is that during a market bubble, during an overvaluation of the market by 20 people sitting online, everybody's nucleus accumbens, this is an average, everybody's nucleus accumbens follows that. It's exuberant. Everybody's nucleus accumbens is going, wow, I really like that. Your nucleus accumbens would do something like that if I injected heroin in you, okay? Or I gave you cocaine. Same thing. It would, if you smoked a cigarette, if I put nicotine in your brainstem, your nucleus accumbens would sing away. This isn't nicotine. This is a market valuation trace. Not only does it track the market, if you go in and you start segmenting the people here, this is a big experiment. Behaviorally, we did almost 400 people. We imaged, the imaging data is from about 50. I think it was 47. You can ask, what does the nucleus accumbens predict? Not only what is it tracking sort of in the present, but what is it predicting in the near-term future? And the answer is, it predicts crashes. So if you look at people with high nucleus accumbens activity, I've divided it into terciles here, the lower one, the middle, and the highest one. So the highest activity in the nucleus accumbens is associated with future price changes, none. The lowest activity is associated with the largest price changes. Let me show you one other thing. If you plot this as a function of crash being a 50% reduction over the next 5 rounds, then the highest level of activity in the nucleus accumbens predicts crashes, okay? So basically, in a population of people that listen to their accumbens in the composite, in a group, are going to tend to be a group that causes a crash because they caused a bubble, right? Bubble goes up, it has to come down. Okay, well, so what? Well, I could tell where they were in the market now because we've been doing other experiments on it by just tracking the nucleus accumbens of these people. Okay, that's not to say this is the only structure that's involved in the valuation and choice in this task. It's part of a whole network of structures. It's just particularly predictive, okay? Don't think that there's just this one spot that's tracking this. There's a network all over your brain that's tracking it. This just happens to be a particularly informative spot. Okay? So now we can ask, what about people that don't get caught up in the bubble? Are they special? And another region of the brain comes in, again, and this is called the anterior insula. So the way to look at this picture here is think of looking through my side of my head, and it's made of plastic. This is the side right here. This is called the insula in green and blue. This is the anterior, the front insula. Here's an eyeball. Okay? So you're looking right here. It's about right here. It's an area involved in the monitoring of your visceral state inside. It's an area involved in gut feeling, right? When you feel unhappy about something, your insula is on. If you're in pain, your insula is on. If I pinch the webbing in your finger, your insula would come on. If I tell you to consciously monitor your heart rate, sit and think about your heart rate and your breathing, this area comes on. Okay? There's a map of your body surface and your viscera represented in nucleus accumbens. It also comes on parametrically when you do economic tasks, and it comes on to the degree to which things weren't going well. It doesn't really, it's reporting on that. So what we think is it's a kind of map at the level of the cortex of your internal body state. It's a cortical representation of your gut feeling, if you will. Let me draw your attention back to the middle here. So this is the nucleus accumbens plot I showed you before. This is the peak of the market I'm showing you here. But now I'm just showing the activity of the nucleus accumbens divided into 2 parts. The accumbens of people that make a lot of money on the task, the accumbens of people who don't make a lot of money. The high guys are in green and the low guys are in red. Notice that the high earners and low earners, they have an exuberant accumbens. Everybody's lower. This is a region of your brain you share with animals that don't even have a cortex. This is a reptile would have a region analogous to this. Okay, it wouldn't have a cortex, but it would have a region analogous to this. Well, that's tracking the market bubble. That's tracking the overvaluation. If I draw your attention over here, the high earners in their insula, the region on the side here, in their cortex, the high earners give a signal in their insula, and the low earners actually drop below, almost drop below 0 right before the peak of the market. So the high earners have, in a sense, a warning signal going off in a region of their cortex, a more evolved part of your brain. And that's associated with, drawing your attention to the left here, this is the most data-heavy slide we have. Again, the peak is here. They're shedding their shares. So I'm showing you the change in the number of units of shares they have here. It's negative. So the green guys are shedding shares. They had a warning signal, and the red guys don't do anything. What does this result in? This results in the guys that listen to their insula make a lot more money, and the market in the group that they happen to be in, and we only recorded, remember, 3 people out of the 20, but those groups are more stable to the way they deal with the bubbles. The bubbles don't get as high. If you throw a bunch of people in that don't have this insular response, it goes through the roof. So the one with 100 that went almost to $150, those are people responding, those are all the lizards, okay? Well, we're all lizards if we turn off our insula, okay? Okay, that's my market bubble. So what does that have to do with the brain and understanding the brain and valuing things? Well, that kind of thing goes on across lots of dimensions. Lots of dimensions. You talk to people, you do what others do, you buy a car like your neighbor. We herd and we group together and we overreact to what others around us do. We're social creatures. And we're now starting to decompose that into signals that we can track, and we can relate those things to mathematical models. Those would have been called just emotional responses before. Now we're teasing them apart into the kinds of things that I've just told you about. Okay, political ideology. Okay, so what do you guys think is going to happen next? Well, he's going to certainly spit the worms out. Those are earthworms, by the way. This is my colleague, John Hibbing, from the University of Nebraska at Lincoln. And this is work he did right after he visited Australia for a long time. Not kidding. John is a political scientist, and he's interested in the biological substrates of political ideology. Okay, so we all know political ideology requires a lot of discussion and deliberation and all the things that make us into political creatures. But what John was interested in was, are there instinctual biological components to it? And he had a reason to think that parts of this were deeply biological and heritable. Okay. Needless to say, he came to me, he asked me would I do a brain imaging experiment, and I laughed him out of the room. He had to work on me for a couple of years. I think political ideology has large heritable like height and hair color and eye color, okay? I think political ideology has large heritable components. I've been studying something at a twin registry in Australia. We've looked at monozygotic twins, identical twins. We've looked at dizygotic twins. We looked at them raised apart and together. And we've given them these political ideology assays, and we get what are called heritability numbers, which is the degree to which you're inheriting the trait, if it is a trait or traits, from your parents, is on the order of 0.5, which means it rivals height. Okay, so that's ridiculous, right? I mean, that just is absolutely ridiculous. I refuse to do the experiment. He's persistent, though. That's how he got all those worms in his mouth. So I was more wrong than he was. I was just wrong, really. He showed me the numbers, and he convinced me that there's some sort of biological underpinning of it. So we did the following experiment with the very pithy title here. Can you notice scientists should never choose the titles of their own papers? We did the following kind of experiment. And let me just give you a quick cartoon story of why it might be that your reaction to disgusting images like that or threatening images might co-vary with the way your political ideology revolved around some central core. Okay? This is just a kind of just so story. So dial the clock way back when we're just coalescing into little bands, maybe in the little villages and whatnot. The way you dealt with two things were crucially important to the survival of the group. The first is contamination, and the second is physical threat. There's no such thing as reacting poorly to contamination or underreacting to physical threat. We don't talk about those people because they don't exist. They didn't make it. Okay? If you make the latrine next to the abattoir, that's a really bad idea. That's just not a good cultural idea. And so lots of cultures, that most we know, learn how to wash foods, clean foods, keep them away from contamination. You also have, as you know, instincts about disgust. If you touch a poopy toilet, if you put your finger in a poopy toilet and you washed my finger off, and my finger touched the surface here, and then I just touched with my clean finger the clean surface where my other finger touched, and I moved it over here, your brain tells you that spot's disgusting. Disgust is transitive. If A is disgusting and you move A over to B, then B is disgusting too, and even if you completely clean it off, C is disgusting. You take no chances with it. If you take a stainless steel bedpan right off of the manufacturing track, right out of the manufacturing thing, and you wash it out, and you put Kool-Aid or punch in it, and you ask somebody to drink out of it, they can hardly do that. It's too important, and the reason it's too important is it just takes one bacterium to infect you. Okay? This is not the way you get polluted by toxins in plants. That goes as the dose. It scales as the dose. So you're willing to touch your mouth to a plant. You do not touch your mouth to a poopy spoon. I mean, any hands for people touching their mouth to poopy spoons here? Okay, so it was a really good idea. So it could be that the way your nervous system instinctually deals with contamination and instinctually deals with physical threat were kind of biological imperatives. And then as cultures grew up around that, then the way in which the ideas and structuring all could have been structured in some mild way by these two biological imperatives. Okay? So it could be that what we call traditional conservatives and traditional liberals on this axis might be predicted fairly well by looking at the way your nervous system reacts to physical threat and contamination. So what did we do? We took a whole bunch of people, 130 people. We show them a sequence of images, emotionally evocative images from positive to negative to neutral. That's a good scale, right? Positive to negative to neutral. Positive, neutral to negative on random time boundaries in a scanner. So they come into the lab. We say, we're doing a visual experiment. We just want you to look at the pictures. So the picture flashes on. I can't show you the pictures because they're from an international affective picture system database, and you're not allowed to show them because it pollutes it basically pollutes the data set, right? You guys would all have seen it, and et cetera. So you sign something that you can't show the pictures. Believe me when I say disgusting image that you saw with John Hibbing, it relates to the disgusting images you would see in the images. So you line the scanner, you see a bunch of random pictures. You get out of the scanner. That takes about 15 minutes. As you're walking over, we sign you up for another experiment. Say, hey, we have this other experiment. It doesn't take very long. Just come to the computer and answer some questions. Okay? And this is part of the Wilson-Patterson. This is the kind of money part. And this is what they do. But it's in a computer, and it comes up one at a time. Okay? So they have a slider bar. They set it between strongly agree, uncertain, disagree, and strongly disagree. Okay, you set this slider bar. And all they are words or word pairs. No explanation. Okay? Now, if you read these, some of these are kind of ambiguous, like foreign aid. I don't know what it means. What does it mean to be uncertain about foreign aid? What does it mean to be certain about foreign aid? I mean, it depends what you mean by foreign aid. Or gun control. In America, this is a trigger issue, right? That's terrible. That's terrible. Are you filming this? I would love to show my wife that. She says I'm the corniest scientist she knows, which is a tautology, by the way. So premarital sex, gay marriage, abortion rights, evolution, biblical truth. And just imagine where you set this. Okay, so what you do is you answer these questions on here. You get a number between 0 and 1. This has been profiled. It's been profiled here and in the US. I don't know how it plays in the UK, right? So the question is, can I take the brain responses to the set of emotionally evocative images, build a mathematical classifier using a modern machine learning technique, and predict your score on this test? And the answer is, I'll give you two answers. The first answer is, it's so good, it's ridiculous, okay? We do what's called cross-validation. So what we do is we look at your brain response to these images, and we go do two things. We find the images in the brain responses that best predict what your score is on the test, number one. And then we go in and ask questions of the images we showed, can we pick out ones that were particularly predictive? Because your whole, lots of areas of your brain go off at once. And at first, we were 100% out of sample. What that means is this. You have a whole sample of data, of images, and you set some aside. And you set the brain responses aside, and you build a model up for the remaining data. And you use that model to try to predict what these guys are going to predict on the score. It's called out-of-sample validation, right? And then we average over that. And we were at 100%. Well, the postdoc, the first author on the paper, was thrilled. And I'm like, we are not publishing a paper where we say we can perfectly predict your score in a political ideology, perfectly predict anything. That's ridiculous. So we hobbled the algorithm a little bit. We got it down to kind of like 99.2. It was such a strong response, averaged over all the images we showed, we can get it with the first disgusting image that you see. Now, the images are randomized, and so the first disgusting image you see might be the first one, it might be the fifth one, the sixth one, but the first one is good enough to predict in the mid-90s, okay? That's amazing. To me, it's amazing because in my mind, still, the answers to these questions and the sense in which these may or may not, these are the social axis. These are particularly predictive here because of their nature. It's social things having to do with family and reproduction and whatnot. They summarize a kind of core feature of who you are. I mean, it seems to be it's an abstract depiction of you in some sense. It's incomplete, of course. And just showing you a couple of pictures, one picture, and using brain imaging, I can guess what your score is on this test. It's even more interesting than that. And these are just regions that lit up. The areas don't matter. Your behavior doesn't know it. The conscious you has no idea. So once we've classified you on these tests and separated you into liberal, moderate, and conservative on this 0 to 1 axis, we can then have you rate the images, okay? Now, the images are already from a normative database that thousands of people have rated before, so we know what the ratings are. And it's kind of a sanity check on whether the people in our group are sort of in the norm. And if you notice, there's no rating difference across these three categories consciously, okay? Threatening stimuli, pleasant stimuli, disgusting stimuli. But your brain completely betrays you. Not only is there a big, huge difference in your brain in terms of how it's responding, it's predictive of what you're going to answer on those questions in the test. Okay, what does this mean? Well, what I'm not going to do is draw kind of simple bromides from this, but what I am going to say is it means that we're getting very good at, not just me, these are just results out of my lab in areas that I find kind of socially interesting. We're getting good at decoding measured brain responses in people in domains that were thought to be too complicated to even wade in. These are early results. The implication for who you vote for, well, actually, the score on this test correlates quite strongly with who you vote for, so I could, that's easy to guess. In this current election, all you had to do was watch that debate the other night to get a sense of what's going to happen. I can't explain the current election, so what can I say? Okay, so the point is, two take-home points. Take-home point number one is modern non-invasive brain imaging is now moving into domains of decision-making that extend beyond a single human body into a market mechanism that you wished you understood in various kinds of ways, and that's just one probe of it, and into an area like political ideology and what it is that composes you as a psychological thinking, feeling being. Where that goes, I don't know, but it is showing you how fast it's developed. You couldn't have done anything like that 10 years ago. And so I just put this last slide up, human-level control through deep reinforcement learning. And I put this slide up because this is from a company called DeepMind that's in London, where my second job is. And it's been bought recently by Google, and it's hiring all kinds of people that I wish I could hire. So people that are computational neuroscientists now are piling into places like DeepMind, and Google's basically snapping up all the scientific talent. But they've been doing things like training neural networks based on the kinds of signals that we see in the nucleus accumbens here and the mathematics we use to capture it to play all the games in the Atari panoply of games, okay? And it beats human performance in something like 85% of the Atari games. And then most recently, this was 2 years ago, and this past year, using the same kind of reinforcement learning algorithm. These are the same kinds of mathematics we use to capture some of the things I've just been telling you. They've trained it to play the game of Go. People know what Go is? Okay. Computer Go playing programs up until 2009 were worthless. Not computer chess playing programs, but Go playing programs were worthless. It's a game that has a lot, the state space of Go is gigantic, okay? They trained a reinforcement learning algorithm with almost no structure. They just presented lots and lots of examples. So the only thing that's changed isn't this mathematics used for it. It's now we have computing power that's cheap, and we can store lots of examples. And so you can train it on a lot of examples. And it beat there isn't really an official world champion, but everybody knows the Korean player is the best player in the world. And he resigned in, I think, 2 hours. Okay, that happened this past spring. What do I think that means? What I think that means is I think computer science approaches, like the ones they're taking here, are clearly going to combine with the kind of stuff I showed you and provide new inroads and insights into the biology of choice and certainly into the way we make decisions, both individually and collectively. And I'm just going to leave you with a few of these final thoughts and leave it right there. Thanks for having me.
Thank you very much, Read. That was very, very fascinating. We can't let you go back to your lab with your 3 scanners without giving you a little token of appreciation from the faculty. Here it is. It's a little bit of a local liquid.
All right.
And thank you again.
Thank you.
Does anyone have a question? We have microphones here.
One mercy question. You have to ask one mercy question.
Raise your hand if you want to ask a question.
Thank you. I enjoyed it tremendously. Are there any obvious differences, like if you use 4-year-olds versus 15-year-olds versus 30-year-olds? And the obvious question is there a big gender difference?
In what?
In your experiments.
Oh, yes. Yeah. Well, the disgusting, we've actually done something like this political ideology assay in kids. We've asked the following question. If indeed parts of this assay are heritable, then you ought to be able to do something analogous in a child while imaging their brain and guess the parent's political ideology when the kids could never even answer questions like that. So that's what we're embarked on. The second thing we're doing is we're doing it subliminally. So instead of flashing the pictures where there's 4 seconds to look at a picture, we flash it for 30 milliseconds and then do something called backward masking so you don't consciously register the image at all. We're very interested in this dissociation that I showed you between what you consciously report and what your brain is betraying by virtue of the machine. So we're actively pursuing that. Thanks for the question.
One more question?
Hey, Reid. In your market bubble experiment with a fixed value of $14 as the underlying, you showed a lot of bubbles. Why wasn't there any deep discounts?
Why wasn't there?
Why didn't the price go below 14?
Why didn't it go below 14? I don't know that. Is that good enough? I don't know. Some of them didn't really bubble. Some of them just sort of rumbled around the fundamental value. I think there were ringers in each group. I think there are people in any group of 20 that kind of recognize the structure of the game and they run the price up real quick and then jump out. So they spin the price up, make sure that there's some other fool that's going to stay on the wheel and then leap off. So I think some of those strategists that you saw there are latent in all those populations. But that's an interesting question to pursue. So there's one here.
One last question. Yes.
Thank you. Have you considered any longitudinal studies whereby you might take young people, children, and run a series of tests on them like you're doing here, and then over a long period of time look at the outcomes, say in mental illness, whether they have propensities to various types of mental illness or behavioural traits or success in the world and so on?
That's a great idea. The question is, what about following children longitudinally doing these sorts of things? I've shown you a small subset of the kinds of things that we do. We used a number of economic exchange games. The market's just one of them. We have another one where you reciprocate with someone else. And we've also shown over the last, we've been doing this kind of thing for about a decade. These economic games are really nice at parsing people out, and they also display boundaries of mental disease states. So we've done it in kids with autism spectrum disorder, ADHD, major depression, something called borderline personality disorder, various brands of people addicted to substances. And we've asked the question, can we use these social exchange tasks? Because what you have to do in any social exchange with another person is you have to imagine their mental state, right? You have to have a model of what they are going to do, and you have to have a secondary model of what they think of you. The only way you do image management is you have a model of their model of you. And we have a project underway in Virginia and in 19 sites through north central London to characterize people at big numbers in the thousands, and we've been embarked on that for about 6 years. So it's something that we're in the middle of. It is slow yeoman's work. It's just a lot of work to do things like that, but we do not have a picture yet of normal mental trajectories through different epochs in life. We don't know what healthy mental living looks like. And so there are a lot of projects popping up around the world to try to characterize that. Thanks for the questions. And do we have one more?
Let's thank. Well, we're running out of
Okay,
time.
right at time.